Welcome students. So today we are moving to the next portion of our chapter 2, plant anatomy and plant physiology. So in this class we are moving to discuss with the transfer section, that is internal structure of dicot root and monopod root. First we will look internal structure of dicot root. Before that, how we will take a transfer section. By using a blade, we will slant cut, we will make a straight cut using the blade, and while we are observing the tissues under the microscope, we can see a picture like this. Okay, so today we are moving to discuss with what are the tissues which is present in this structure and how they are arranged inside the system. First, the internal structure of dicot root, that is beam. Example is B. So, transverse section of dicot root having structures like epiblema, cortex, endodermis, finally stele. Epiblema, cortex, endodermis and stele are the major structures which is found in the transverse section of dicot root. Okay, first one is, so this is the transverse section of a root. This is the ground plant. If we take the slide cut, if we take the cross section of the root and we observe under the microscope, we can see a picture like this. If we take the cross section of the root and we observe under the microscope, we can see a section like this. Okay, and this is a portion which is enlarged. We are enlarging a portion from this ground plant and we are drawing it in enlarged portion so that we can study in easy manner. Okay, so this is the ground plan and this is the enlarged view. So what is ground plan? Ground plan means we are cutting the root in a circle shape and we are observing that section under the microscope. So we will observe picture like this. And this enlarged portion means we are taking a small portion of this. We are taking a small portion and we are enlarging. We are drawing in large view so that we can study in easy manner. So epiblema. What is epiblema? Epiblema is the outermost layer of the internal structure of dicot root. So outermost layer of the section is called epiblema. Okay, so there is no cuticle and stomata. So absence of cuticle and stomata are in the epiblema, and there is presence of unicellular hairs. Unicellular hairs are present in the epiblema. Okay, it is also known as rhizodermis or piliferous layer. So epiblema is the outermost layer and it lack of cuticle and stomata. Presence of cellular hairs like root hairs can be found, and also it is known as piliferous layer or rhizodermis. Okay, so this is epiblema. So next, inner to epiblema, it is called cortex. So cortex is the multi-layered large zone. Multi-layered large zone. This is the cortex. It containing so many cells which are arranged in multi-layered form. Okay, and these are thin-walled parenchymatous cells. Thin-walled parenchymatous cells are arranged in multi-layered large zone. Okay, and the cells are not tightly arranged. The cells are arranged very loosely. So it has intercellular spaces. Intercellular spaces means space which is present between the cells are called intercellular spaces. So cortex, it is the inner to epiblema. We will see cortex. Cortex is thin-walled parenchymatous tissues having intercellular spaces. And the function of cortex is it stores food and water. It stores food and water. So first layer is epiblema, inner to it is cortex. Then next is endodermis. So inner to the cortex it is called endodermis. Okay, actually I am taking some portions from the main picture. Look, this picture. Actually I am emphasizing the each portion. This is the epiblema, this is the cortex region, and together this is called the endodermis region or stellar region. Okay. So this is the endodermis. Innermost layer of cortex. Okay, we know cortex is just below the epidermis. So the innermost layer of cortex is called endodermis, and it is made up of barrel shaped cells and are closely packed. Made up of barrel shaped cells and are closely packed. There is no intercellular spaces between the endodermis cells. Okay, and some band like thickening can be found in the endodermis. Some band like thickening can be found in the endodermis. Look, this light pinkish color outer layer is called endodermis. Okay, so this is cortex. The innermost layer of cortex is called endodermis. This light pinkish color called endodermis. It having some thickening called Casparian stripes. Okay, so the band-like thickening that is found in the radial and inner tangential wall are called Casparian stripe. But Casparian stripes are present in almost all the endodermis cell, but some cell it lacks the Casparian strips that are called passage cells. These dark in the light pinkish cells, some dark color pink you may note here one, here one, here one, and here. Okay, so those are called passage cells. So endodermis cells which lack the Casparian thickening are called passage cells. It helps in the movement of water and dissolved salts from cortex to xylem. So here xylem it containing so many water bodies as well as dissolved salt. Okay, dissolved salt means the minerals which are dissolved in the water that are called dissolved salts. So the water and dissolved salts from the cortex it enter into the xylem through these passage cells. Okay, so these are endodermis. Then next one is stellar region. Finally, the middle region is stellar region. So all tissues which is inner to endodermis. So this light pink colored circle is endodermis, and inner to there is a dark pink colored circle. It is called pericycle. So all the layers which is found inner to endodermis is called pericycle. So inner to endodermis is called stele, and the layer of tissues which is found just below the endodermis is called pericycle. So it is the site of origin of lateral roots. So lateral roots are developed from the pericycle. Then inner to pericycle, this is called vascular bundle. Vascular bundle have vascular tissues. We already studied what are vascular tissues. Yes, xylem and phloem. This is xylem, and these blue colored tissues are called phloem. Okay, so vascular bundle, it is radial. Radial means the xylem and phloem are not in the same radius. Xylem is in this radius and phloem is in another radius. Okay, so xylem vascular bundle is radial arrangement, and the xylem is X arc and tetrarch. Xylem is exarch and tetrarch. What is exarch? Exarch means the protoxylem that lie towards the periphery. Protoxylem means small xylem. This is the small xylem, and it is faced towards the periphery, that is the outer region. And the central big xylem, that is metaxylem, which is present in the center. So it is, this condition is called exarch. Okay, and tetrarch means there is 4 layers of xylem. 1, 2, 3, 4. So that is tetrarch. Okay, and the tissue which is present between xylem and phloem is called conjunctive tissue. These are xylem, this is phloem, this and finally these 4 are phloem. So the tissues which is present in between xylem and phloem, these white colored tissues which is present between xylem and phloem are called conjunctive tissues, and it is made up of parenchyma. Then finally pith. Pith which is present here. Pith means below the endoderm, below the stellar region we can find some pith region. So young root containing pith. Normally young roots containing pith, but when the root become older the pith will be absent. Okay, so these are about the internal structure of dicot root. Example is bean. Okay, now we are moving to what is the internal structure of monocot root. Internal structure of monocot root. Example, maize. So the transverse section of monocot root having structures like epiblemma, cortex, endodermis and stele. Okay, so Just like Dicot, all the characters like epidermis, cortex, endodermis and stele is found in the internal structure of monocot root, that is maize also. So this is the ground plan of monocot root and this is the portion which is enlarged. Okay, so now you know these are called epidermis, that is the outermost region, outermost layer of the section are called epidermis. It containing root hair, it is also called as piliferous layer. Then inner to epidermis, it is called cortex region. Multi-layered thin walled parenchymatous cells are loosely arranged. This portion is called cortex. Then inner to the cortex, innermost layer are called endodermis. The light pinkish color are called endodermis. Endodermis are made up of Casparian stripes, but some cells are devoid of these Casparian cells that are called passage cells. This dark colored 1, 2, 3, 4, 5, 6, 7, 8. Okay, so these pink colored cells having dark pink colored cells that are called passage cells. So through these passage cells the water and dissolved salts can be moved from cortex into xylem. Okay, so inner to the endodermis it is called pericycle, then inner to it is called the vascular bundle. Okay, same as dicot root but some change, same as dicot root but having some changes. So first one is epidermis, outermost layer with thin walled parenchyma, same as dicot root. It absence of cuticle and stomata, having no cuticle and stomata. It is also same as dicot root. Then root hairs help in absorption of water and minerals from soil. Okay, both dicot root as well as monocot root, the presence of root hair which helps in the absorption of water and minerals from the soil. And it also protects the inner tissue. Actually, what is the function of epidermis? Epidermis or epidermis, it mainly having the protection function. It will protect the inner tissues from any damages. So next one is cortex. Below the epidermis, it is called cortex region and it is a multi-layered large zone made up of thin-walled parenchymatous cells having large intercellular spaces, which is same as dicot root. Okay, and it helps to store food and water. It helps to store food and water in monocot root. Then next one is endodermis. Endodermis is the innermost layer of cortex with the Casparian strips and passage cells. We already discussed what is endodermis and what are Casparian strips and what are passage cells. Okay, we know Casparian strips are made up of suberin. Casparian strips are made up of suberin and it is impermeable to water. Suberin cannot permit water into the vascular tissues. Okay, so that only water can move into the stellar region through passage cells. Passage cells are endodermic cells which devoid of suberin thickening, that is Casparian stripes. Okay, then next one is stellar region. Next one is stellar region. So inner to the endodermis, this region is called pericycle, that is the dark colored ring is called pericycle, that is inner to endodermis. It is a single layer thin walled cells. We already discussed what is pericycle. It is a single layer thin walled cells where lateral roots are produced. So it is a compulsory 1 mark question, from where the lateral roots are produced. It is produced from the pericycle. Then vascular tissues. It consists of many patches of xylem and phloem that are arranged radially. Okay, so here also the xylem and phloems are not in the same radius, so it is called radial arrangement. Here here also the vascular tissues are exarch. So in root the xylem is exarch condition. Exarch means the protoxylem which is faced towards the endodermis, that is towards outside, and whereas the metaxylem which is towards the center. Okay, and how many xylem tissues are there? 1, 2, 3, 4, 5, 6, 7, 8 tissues are there. So it is called polyarch. It is called polyarch. Okay, so in dicot root we said it is tetrarch, but in case of monocot root it is polyarch. Then the conjunctive tissues which are made up of sclerenchyma cells, but in case of Dicot root, we already discussed that the conjunctive tissues. Conjunctive tissues means the tissues which is present between the xylem and phloem are called conjunctive tissues, and it is made up of parenchymatous cells. But in case of monocot root, it is made up of sclerenchymatous cells. Okay. Finally, pith. Pith is present in the center. The center light blue is pith. It is made up of parenchyma cells with intercellular spaces. The parenchyma cells which are not closely packed, it is loosely packed and having intercellular spaces. Okay, and it contains starch grains and it store food. Pith contains starch grains and it store food. So these are the internal structure of monocot root and dicot root. I think you got the idea about this. You go through the textbook, and I am giving you a homework that you have to prepare the difference between dicot and monocot root. Difference between dicot and monocot root. Okay, so in dicot root we already discussed pith is present only in the young stage. At older stage there is no pith. But in case of monocot root, pith is present in both the phases. That is, pith is present in older root as well as young root. Okay, and the xylem, we already counted the number of xylem patches which is present in dicot root. It is 4, that is tetra, but in case of monocot it is polya. So just like that you have to prepare the difference between dicot and monocot root. How you will prepare? First you have to study the internal structure of dicot root and internal structure of monocot root. Then you yourself want to prepare the difference between dicot and monocot root. Okay, so these are up to today. Thank you for your patience.